Students, welcome to our osseous tissue histology review. What we're going to do is go over the compact bone slide or the slides and the spongy bone slides just as a quick review so I can point out some of the items that we've asked you to identify in your lab activity. Before we do that, I just want to draw your attention to the picture here up in the upper left-hand corner. That is the lowest magnification, and within the blue block is the area that we're looking at. So it gives you some perspective so we don't have to look at every single slide. You can't really see much in this view right here, can you? You have to really zoom into the blue box right here in order to see some of those features. So this is what it would look like if you were on the histologyguide.com website and were to zoom into this area here. So if we go through the items, we'll just go through them one by one based on your lab activities. One of the structures that we ask you to identify is the individual functional unit of compact bone. An individual functional unit means the smallest unit within a structure that has the ability to carry out all the functions of the organ or larger structure itself. So circled in red there is an osteon, and you can see several osteons here. All these circular structures, like I mentioned in the lecture, and I think they mentioned in the textbook, that these look an awful lot like cross-sections through tree trunks. So in red there, those are the osteons. In the center of the slide, you'll see this space right here. It's not an empty space. That's the central canal, or what we call the haversian canal. And what we find in there are blood vessels and nerves traveling up the osteons. Remember, osseous tissue is living tissue. It's got living cells in it. It needs to have oxygen carried to it, nutrients carried to it, and carbon dioxide and other waste products removed from it. So traveling up through those central canals will be blood vessels and nerves. Now surrounding the osteons, or in between osteons rather, are a feature that sometimes students have difficulty identifying. And these are what we call interstitial lamellae. So you can see between the osteons that there's osseous tissue, but it's not arranged in an osteon. So you wouldn't have like empty spaces between those cylindrical osteons. You still have osseous tissue, you still have osteocytes, you still have that cement-like ground substance, but it's not arranged in an organized fashion. So what's highlighted there in green, those are called interstitial lamellae. And then another feature we're asking you to identify are what we call concentric lamellae. Concentric means with the center. So these are the lamellae that surround in rings that central canal. So think of concentric lamellae just like the growth rings on a tree trunk. I'll go ahead and point those out on this view, and then we'll move to the next slide, which is a more magnified view. So I'll use as a highlighter, I'll use the blue, and I guess we'll go for this one in the center. So these little rings of growth, I'm going to skip one here so you can just see the difference. Those rings of growth are called concentric lamellae. So I'll just number them from the outside in. So here's one, Here's one in gray. This is another one in blue. There's another one. And in gray, again, there's a fourth one. Those are what we call concentric lamellae. Again, it's osseous tissue, but osseous tissue that's arranged in concentric circles around the central canal. Let's go ahead as well on the previous. No. Let's see if we can add one here. Or not add one, but I'll add a slide here, and we'll see if we can get a better look at a perforating canal. And look at the next slide. Here we go. This is really, really nice. Again, concentric lamellae are going to be, I'll do this first one right here. I'll skip one so we can see the difference. There's another one here. Skip another one. So these concentric rings of osseous tissue. For a point of reference, here is that central canal, the blood vessels and nerves traveling up it. And then another thing that we're asking you to identify are the lacunae. And the lacunae are these little black dots. And those are going to be spaces where we find osteocytes. Now, in this particular slide, like in your lab activities, it says that this piece of bone has been, the way it's been prepared is it's been heated to really high temperatures, and that removes all the organic components, the carbon-based components. So the cells, they're organic, so when they process this tissue, the cells dissolve, and so do the collagen fibers. So although these structures appear as though there's something black inside of them, they're actually spaces that are now just filled with ash. What would normally have been inside would be an osteocyte. You know that osteocytes exist within lacunae. So that's what we would normally find there, but we're just asking you to identify the the lacunae and not the osteocytes in this slide, because there technically aren't any. And then the canaliculi. The canaliculi are, let's do green here. The canaliculi are these little black streaks that you see. And those are microscopic passageways within bone. And what they're going to allow for are oxygen and nutrients to travel down those pathways into the osteocytes that would be in those lacunae. So nutrients and oxygen are going to pass from that central canal and make their way out to those osteocytes. And the same is true for waste products. Waste products are going to leave those lacunae from the osteocytes and make their way back into the blood vessels in that central canal. A perforating canal is not visible in this slide. Let's go back and see. Okay, here we go. This is not the same slide that you see from histologyguide.com, but it's a pretty good example where, let me orient you here first, where here is an osteon. Here would be its central canal, and then off to the side, this black streak right here, that's what we call a perforating canal. And this is a longitudinal section through a vessel, a blood vessel that's passing through the tissue itself. So we're only seeing a part of it just because the knife didn't graze the whole thing. But imagine that that blood vessel perhaps even connecting to the central canal of another blood vessel, a central canal of another osteon. So these perforating canals, they are blood vessels that travel through the bone, carrying nutrients from one place in the bone to the next. Okay, so that is all for the this slide. Let's move on to the next one. And this is also compact bone, but this is compact bone that's been prepared differently. In this case, what's been removed are all the inorganic components, the minerals, the calcium, the magnesium, the phosphate salts. What's been left behind are the osteocytes and the collagen fibers. The collagen fibers that make up the periosteum, the collagen fibers that are actually embedded in the osseous tissue itself. So this donut-like structure is a cross-section through a long bone, right? So there isn't too much that we can see in this view. imagine there being a whole bunch of bone marrow. Remember, bone marrow is made of red blood cells and fat, as well as scattered white blood cells. So those cells would be in the center. But if we increase the magnification, and let me move this bar out of the way here so we can see kind of what we're looking at. Okay, so if we this one first. One thing I didn't mention in this larger view is this space right here. That's the medullary cavity. And I did mention that that's where you find bone marrow, but I didn't mention the name of the cavity itself. To the next magnification, this is what we see. And let me move this slide over so we can see that. The view, the larger view. So this is what we were looking at before, right? This is our previous view, our cross section through a long bone, and we've zoomed in now to this little area that is represented by that black box, okay? So we'll go ahead and move that back over. And what we see there, kind of smack dab in the center, again, is an osteon. Right here. You can see part of another one here, right? And in the center of that osteon is a central canal. The lacunae now are what you see here. These are the little pockets that have the osteocytes in them, the mature bone cells. So this one's a little bit difficult to see, but you can see that black dot right there. That's the osteocyte itself. Okay, so those osteocytes are packed into the lacunae, or the lacuna. Lacunae is plural, lacuna is singular. The lacuna there has an osteocyte on the inside. Blood vessels you can barely see. There would be a blood vessel in that central canal. Here's a blood vessel over here. And then, let's see, go back to this. So we still have osteoblasts to identify. So what we're going to do is, go next time, move on to this view right here. And remember where we find osteoblasts. We're going to find osteoblasts in the cellular layer of the periosteum, but also in the endosteum. So we'll move this around and look where we're looking at the cross section of the bone now. We're looking at a larger section here, but also this section includes where we would find the endosteum, that lining of the medullary cavity. So we move this back over. Here's the medullary cavity right here. And it's a little bit difficult to see. I think I have on the next slide a little bit more zoomed in view, but that's an osteoblast. That's an osteoblast. And then also we would find osteoblasts here because they're going to be in that inner cellular layer of the periosteum. This is the outer fibrous layer of the periosteum. So let's go to the next slide. Here we go. This is a really good one. So now what we've done is, let's look and see what we're looking at. We're looking at the edge of this cross section through compact bone. And here you see the osteoblasts. And notice they're not in lacunae. Osteoblasts do not exist in lacunae. Only the mature bone cells, the osteocytes. Okay, so that makes this then the inner cellular layer of the periosteum. And then that makes this, that makes this the outer fibrous layer of the periosteum. Right there. Okay, so now we can see the other location where we find osteoblasts, and look where we are. We're focused now on that lining of the medullary cavity. And this is what I was trying to show you before. It was really difficult to see. But there's an osteoblast Here's an osteoblast, although it's not very dark. There's an osteoblast. So there aren't many, but let's just kind of imagine that there would be osteoblasts here, right? Just draw those in. Okay, and then let's move on to spongy bone, sometimes called cancellous bone. Again, this is osseous tissue, but remember with spongy bone, the arrangement of structures is very different because the function is a little bit different. So the function of spongy bone, remember, is to take forces from many directions or be able to handle stress from many directions. So you don't see the organization of structures within osteons. It's more of a random arrangement of osseous tissue going in many different directions. So this is the view from histologyguide.com. Here you're looking at a section straight through a long bone, and that's on this one right here. This one is part of a cross section right here, and then this is a longitudinal section. We're just going to look at the cross section here. Okay, so again, not a whole lot that you can see on this magnification, but I do want to bring your attention to some of the things we can see. This would be the fibrous layer of the periosteum right here. It's kind of coming away, but that's probably just an artifact of preparation of the tissue. The cellular layer of the periosteum would be right here. Right, and you just can't see that. And then what else you see would be, let's do red here. This is all the compact bone portion right here. And then what lines the medullary cavity is spongy bone. So this looks a little bit different than when we were looking at the cross-section through a long bone, because we were looking at that in the compact bone slides we were looking at. That was a cross-section through the diaphysis, where you don't have a whole lot of spongy bone. This would be a cross-section through an epiphysis. So all of this area right here would be the center of that bone, right? And then that would also be the medullary cavity. But because this section has been taken from the epiphysis of a long bone, you see much more spongy bone there. So let me good, we can erase that. And then on this view, I want to point out the trabeculae to you. So these little beams of tissue, these are the trabeculae. And trabeculae is just the name for these struts of spongy bone. So all of this. We can come across here too. These are the trabeculae. So what we really want to look at are the trabeculae, because we want to be able to identify osteocytes, lacunae, and the endosteum. From this view, you can't really see it, but know that because this is the medullary cavity, you're going to have marrow, which is made up of red blood cells, white blood cells, and fat, right? So you'll have some adipocytes in here. Let's go ahead and look at it on a higher magnification. So again, in this view, you see the periosteum. This would be the outer fibrous layer right here. You can kind of see it peeling away, but again, that's because of the way that the slide was prepared. You can also see the central canals, because this is the compact bone portion of this section, right? Where the spongy bone is in the center. So I'm just pointing out, giving you some landmarks here so you know exactly what you're looking at. And then here are the trabeculae. I'll use a highlighter this time so it's easier for you all to see. These are the trabeculae. right? And then if we, again, oops, between the trabeculae is where you're going to find the marrow, red blood cells, adipocytes, and we'll just draw in a few white blood cells. This is exactly the same slide, exactly the same magnification. So let's continue. Here we go. Let's look at where we're looking now. So, same slide, but now we are really zoomed in on one of those trabeculae so that we can see some of the other structures we've asked you to identify. So first things first, I'll highlight it. This would be the endosteum. Right, that's going to line the trabeculae, and remember the endosteum is made of osteoblasts and osteoclasts, not osteocytes, but osteoclasts. And the osteoblasts within that endosteum, let me erase that. These are the osteoblasts of the endosteum, right? Those little black dots. Another structure we've asked you to identify are the lacunae. So here is a lacuna. Here's another one, just barely caught by the knife there. Here's another one. And within that little black dot, that is the osteocyte. That black dot right there is the osteocyte, and that purplish dot right there is the osteocyte. Okay, same slide. We just moved around a little bit. Now we're looking at the outside, right? And I showed it to you in an earlier slide, but again, I want to make sure that you understand that from here to here is the periosteum. It's got two layers. This would be the outer fibrous layer of the periosteum, and then this would be the intercellular layer of the periosteum. And you can see the cells there, right? What type of cells? Those are osteoblasts. Another thing I can point out on this, and we've seen it before, are the lacunae, which are those little white dots, and the black dot inside is an osteocyte. And then also, we will not be looking at the slide entitled endochondral ossification. It says at the top of your lab activity, check with your professor whether they require this slide. I don't require the endochondral ossification slide. Okay? All right, that's all I've got for you. Hope this helped. See you next time.